Let's go to our first panel of the day on this track. And I'm going to bring on Dr. Jason McCarthy, who is the Senior Managing Director at Maxim Group. And I'll let Jason introduce the panelists and take it away.
Great. Thank you. Welcome, everybody. The topic of this panel is really, getting back to fundamentals post-COVID. I don't know if we're really post-COVID yet, and we're going to talk about that a little bit as part of the GeoVax story. But I'm pleased to welcome BriaCell and Bill Williams, CEO, GeoVax Labs, David Dodd, who've known for a very long time, CEO as well. Same thing for Steve O'Loughlin, CFO of Actinium Pharmaceuticals. I've known these fellows for quite some time, and they've done some incredible things in the oncology space. And that's really, where I want to start this discussion today, and that's getting back to fundamentals. So COVID was all of 2020. Like I said, we'll talk about that with David in just a second. But in oncology, we're really starting to get back to basics and back to fundamentals. And I'll start with you, Bill. Can you talk just a little bit about where you see the oncology space moving towards today, and maybe dovetail that into what BriaCell is working on?
Yeah, I think there's a few things there, Jason, that are at the same time concerning, but also opportunities. One of the things that happened in the past year is that a lot of patients did not come in for checkups. And unfortunately, I think that presents us with a situation where oncologists are going to be seeing patients with more advanced types of tumors. And I think that is going to have an impact on the biotech environment going forward in the next couple of years, as people start to realize things got let go that shouldn't have gotten let go. At the same time, we're seeing tremendous advances in things like immunotherapy, where we're harnessing the power of the patient's own immune system to attack and destroy cancerous tumors. And this presents a huge opportunity for these therapies to be developed, especially in patients with advanced types of cancer. So I think that there's at the same time an opportunity here for therapies that work especially in advanced cancers, and for those that take advantage of the patient's immune system, as well as those other targeted therapies and similar therapies that are showing great advantages over traditional chemotherapies.
Right. Let me just jump over to Steve, because we could talk about immune therapy broadly from the work that Bill's group is doing. David's group is in MUC1 on the cancer vaccine side, but everybody looks at actinium around Sierra and bone marrow ablation. It's a phase 3 trial. We could talk about that, but you are doing some work in immune therapy with CAR-T and leveraging your technology towards that. Can you talk a little bit about what actinium is doing in that space that seems to be kind of flying under the radar for you guys?
Yeah, absolutely. And maybe just a quick overview on the underlying technology. So we're advancing clinical stage antibody radiation conjugates, which are able to take a proven therapeutic modality, radiation, that's been used for decades and is still used today to treat over half of all patients with cancer. And we're delivering it in a unique and different way inside the body at precise cellular levels. In the case of our clinical programs now, they're antibody-directed radioisotopes or antibody radiation conjugates. As Jason mentioned, we're using our technology now to condition patients not only for bone marrow transplant or stem cell transplant, but also CAR-T and adoptive cellular therapies, as well as gene therapies. And with our technology, we're able to target certain immune cells and deplete them to create an ideal immune environment, homeostatic environment for these cells to then persist and expand and exert their therapeutic effect. So it's an exciting evolution of our portfolio, and very much looking forward to providing updates on some of the ongoing trials, including our collaboration with Sloan Kettering, where we've been jointly awarded NIH funding.
David, you guys have done tremendous things in infectious disease, but I think something that gets less noted with the investment community is that you do have a whole oncology component to GeoVax, and in particular around the MUC1 antigen, and in immunology circles and on Wall Street, everybody knows MUC1 as it relates to CAR T. It's been for TCRs. People have gone after this, but you're actually working out, with the University of Pittsburgh, and that's where the MUC1 antigen was discovered. Can you talk a little bit about that?
Sure. It is a newer area, but we're applying the exact same technology we utilize for constructing vaccines against infectious diseases. So we're fortunate to be working with Oya Finn at University of Pittsburgh, who was one of the first, if not the first, to discover and characterize the MUC1 tumor-associated antigen. And we've had some very promising results in animals, in terms of both a therapeutic as well as a recurrence model. And essentially what we do is we construct a tumor-associated antigen vaccine with our MVA VLP technology, and we're also looking at cyclin B1 and E6, E7 antigens. But with MUC1, we do that. We combine it with an immune checkpoint inhibitor, and what we've seen is it enhances the utility and the performance of the checkpoint inhibitor in both situations, and all beyond just simply the checkpoint inhibitor alone or our vaccine alone. So if we construct a MVA VLP MUC1 with a peptide, we get some positive response, but it isn't near as good as when one does the combination. And if one just does the checkpoint inhibitor, it does not perform as well as when it's combined with that. So we're currently targeting to move that into clinical development in conjunction with Pittsburgh and Dr. Finn, probably within the next 15 months.
And Bill, so David had mentioned tumor-associated antigen, the BRIA IMT approach to, it's a little bit different from what David's group is doing, but what you're doing in breast cancer with multiple antigens, multiple HLA types. Can you talk a little bit about how that technology works? And can you do this with or without a checkpoint inhibitor? Do you need one for the approach you're taking?
Right. So to go backwards in the questions, we can do it with or without a checkpoint inhibitor. But of course, our types of therapies, David's as well as ours, puts the foot on the gas of the immune response. And so you're making a targeted and stimulating a targeted response against the tumor or the tumor antigen. And the checkpoint inhibitors take the foot off the brakes. So it just makes sense to use them together, that you're going to get some synergy there. But in terms of the way our therapy works, what we have is at the present, a breast cancer cell line that also functions as what's called an antigen-presenting cell. In other words, it is able to not only provide breast cancer proteins, or also known as antigens, to the immune system to recognize and be stimulated to attack the patient's own tumor, but it also is able to act as an antigen-presenting cell. Those are the type of immune cells that start immune responses and boost immune responses. And so we believe that our therapy is able to not only stimulate an immune response, but actually also to boost it markedly with this particular mechanism of antigen presentation. And this is all dependent on what's called the HLA type of the patient. HLA molecules are the molecules that start immune responses, but they're also polymorphic. In other words, they're different in different people, but then they're shared by some people. So for example, I see on this panel right now, 2 of us share bald heads and 2 have hair.
The 2 better people, by the way.
Yeah. So that's
just saying,
polymorphic.
throwing it out there.
Yeah. HLA molecules are kind of like that you can share it with another person, or it could be different. Eye color is the same way. And so, what we've noticed is that if the patients match our cell line at a couple of HLA types, their response rate is much higher. So now we've gone on to engineer our cell line to express multiple HLA types. So we'll be able to personalize the therapy, and yet it's still going to be off the shelf. We don't have to do individualized manufacturing for each patient.
And breast cancer has been an incredibly tough area to have immune therapies actually be effective. We saw a lot of stuff coming out of San Antonio in December with Celos, and I think it's Greenwich Life Sciences had some tremendous vaccine data. Going back to vaccines, David, is breast cancer something that's on your radar for MUC1, or where do you think you and the University of Pittsburgh might try to target first, or do you just kind of do a basket study to see, where the best indication may be?
We haven't settled on that, on what the initial indication is going to be. That is a topic of continuous discussion right now, both internally and with our advisors. And so it's interesting that earlier today I was having that same discussion with people internally in here. So we haven't settled on it, but it'll be a solid tumor, and we just haven't yet resolved which one it's going to be.
Got it. And then I guess when all else fails and you progress to a very severe point in your disease, Steve, this is where actinium can be very effective for patients, hopefully, right? You have IOMAB in a phase 3 trial. Can you talk a little bit about that trial in AML? And also, I don't know if you can maybe articulate a little bit or opine on how using bone marrow ablation, getting these people just a little bit healthier, surviving longer, maybe a vaccine could come in behind IOMAB BMT.
Yeah, sure. So IOMAB-B is intended to condition patients for bone marrow transplant, initially targeting patients with relapse refractory AML, patients with active disease, in some cases as high as 30% blast, which is a patient population that today is typically not considered for transplant. We licensed IOMAB from the Fred Hutchinson Cancer Research Center. It had been studied in several hundred patients, multiple hematologic indications, multiple clinical trials. But really, there's a significant unmet need in relapse refractory AML. Patient survival is typically measured in only a couple of months. The beautiful thing about IOMAB-B is it targets something called CD45. It's a cell surface protein that's expressed on blood cancer cells, leukemia and lymphoma cells, as well as normal nucleated immune cells. So with a single therapeutic dose of IOMAB-B, we're able to simultaneously ablate the patient's cancer, their underlying leukemia, as well as their immune cells, to put them in a position rapidly, in just a matter of days, to receive their bone marrow transplant or their stem cell transplant. And what we've been able to demonstrate thus far in our phase 3 trial is that all patients who have received the therapeutic dose of IOMAB have gone on to successfully receive their bone marrow transplant and have engrafted without delay, which compares favorably to what we've seen in the control arm of the study. But Jason, yeah, to your point, there's a lot of work that's being conducted now looking at patients post-transplant, looking at additional therapies that can be given to patients to kind of, keep their disease at bay beyond what you would hope to see after a transplant, where you get a graft versus tumor effect, where the patient's newly reconstituted immune system is actually recognizing their underlying disease and is continuing to then attack it post-transplant. So, there's no doubt that we continue to be able to have to make advances in patient cancer care. And I think combination therapies will be the way of the future, and certainly integrating that with immune therapy, I think will be very significant.
So a question first for Dave, and then Bill, you can follow up. Relatively similar technologies. Are these areas that you would consider what Steve is mentioning? You have bone marrow ablation, and you reconstitute the immune system with a graft, and that's where we always thought, or at least my group, that vaccines may be most effective, or immune therapies where you've debulked more or less. And so, David, can you talk a little bit about maybe the potential of moving into an area like that, just based on the technology that MVA brings and your VLP? So maybe starting with the latter first, and why that might make sense for you.
Yeah, we've had promising data, albeit animal data, in recurrence models, and we think there's a real role there that people are treated, they seem to be fine, but you need to be able to reduce the risk and likelihood of recurrence. And we've generated some very promising data in models related to that. And the value of the MVA as our vector platform, as you know, is it's a very large virus. It has multiple encoding sites, so we're able to encode multiple antigen or virus proteins in it versus other technologies of vaccines, mRNA, adenovirus. You can pick one virus protein at a time to load it up. We've loaded up 4 or 5 different ones, so we'll do a lead protein, and then we'll do other structural proteins that'll give you a greater either durability or greater memory, et cetera. So we generate both antibody and T cell responses, and we think that makes our technology very applicable in the area of immunotherapy.
Bill, where do you see your technology? Breast cancer, is that, I mean, it's not AML like where Steve's group is, but would the patient have undergone surgery, whether lumpectomy or mastectomy, and you're in a similar setting per se?
Right, and I think that you're right that traditionally it's been thought that you need to use, what so-called therapeutic cancer vaccines, or what I like to call targeted immunotherapies, that they need to be used in patients who have a large tumor bulk, and that the immune system wouldn't be as suppressed in that situation. I think what we're learning more recently is that the immunosuppression that happens sometimes is very localized to the tumor. So, for example, with PD-L1 expression, which of course is the target of Tecentriq and molecules like that, those tumors can locally express tumor-infiltrating lymphocytes that are there. So the immune response is there, but the tumor can still shut it down. We've been very fortunate with our technology to have success even in very late stage patients who have failed multiple prior therapies, including some with relatively bulky tumors. So I think that the paradigm still exists to some extent, that when you have a healthy immune system, obviously you're going to be better off. But as we learn more about what are the signals that are shutting down the immune system, when we can block those, then it opens a whole new vista of the use of these immunotherapies, even in patients with very advanced disease.
Right. So now that we have an understanding of the backbone technologies of what what your companies are working on, perhaps because this is about fundamentals, right? So we're why do you want to take a look at what these companies are doing, and a lot of that is going to be driven by what's coming up in the clinic, your catalysts, and things of that nature. So Steve, being the most clinically advanced company right now in the group, you are a pivotal stage company, Actinium.
Yeah.
Maybe you could talk just a little bit about where that Sierra trial is and what we've seen at the prior 2 ASH meetings, obviously one live, one not, but good data, right? So maybe you can help us understand where the program is at.
Yeah, absolutely. So IMAB is in the pivotal phase 3 Sierra trial. It's the only randomized phase 3 trial to offer bone marrow transplant as a treatment option for these patients, with transplant being the only potentially curative treatment option. We're over 75% enrolled in the study, and we've presented 75% safety and feasibility data. Jason, as you mentioned, at ASH just last December, also earlier this year at the Transplantation and Cellular Therapy Conference. What we've demonstrated thus far, and as I mentioned just a little while ago, is all patients who have received the therapeutic dose of Iomab-B have gone on to receive their transplant and engrafted without delay. In the control arm, which is physician's choice of salvage therapy, as there is no standard of care in this patient population, unfortunately, only 18% of patients have gone on to transplant and engrafted. Put another way, 82% of patients on the control arm have failed or are failures for the primary endpoint, which is 6 months durable complete remission. But in order to get there, you have to first achieve a remission. In addition to the transplant data that we've been able to present, we've also demonstrated that there's a lower rate of non-relapse transplant-related mortality in the Sierra arm, lower rates of severe mucositis, sepsis, and febrile neutropenia. The therapy has been well tolerated. It's given as a single infusion And then patients, as I said, several days later are able to go on and receive their bone marrow transplant. These trends have been consistent at the data updates we presented at 25% enrollment, 50% enrollment, and now 75% enrollment, consistent with the profile that we saw in the data that was generated out in Seattle at the Fred Hutchinson Cancer Research Center. So now our attention is on completing enrollment of the Sierra trial, which we expect to occur this year, so that we can present top-line data for that primary endpoint, 6 months durable complete remission. So, very excited to be in this position. We've been strengthening the team. We were able to strengthen the balance sheet last year, so we're now fully funded through completion of the trial top-line data.
Right. So I want to jump over to Bill and back to you, David, after, because we need to get into the COVID work that you're doing, which I think is tremendous. So Bill, where are things, a couple of questions, where are things in the breast cancer program? Kind of what are the next steps and what we could expect? And maybe you could touch on just a little bit about the PKC work, the PKC delta work that you're doing, that's touching on RAS pathways, and KRAS has been a very, very hot subject for the last 18 months or so with groups like Mirati and Revolution. So maybe you can walk us through a few things that the company's doing right now.
Sure. So for our lead program, BRIA IMT, we're in collaboration with Incyte Corporation using their PD-1 inhibitor, retifanlimab, which is very advanced in some other indications right now. And we're getting some very intriguing results there. We're continuing to enroll that study, and the goal is to get a dozen or two patients data's worth in third-line or later breast cancer where there is no approved therapy. And then to go down to the FDA and negotiate with them what's called a special protocol assessment, or an SPA, which is basically the design of your registration study. And the registration study is the study that you do to get your drug approved. It's the one that Steve's involved in right now, he was just talking about. And so our goal is to get that agreement from the FDA as to what that study design is going to be, because then it'll be very clear exactly what we need to do to get the drug approved, how much it'll cost, and what the target patient population will be. That's the point at which we would hope, and where we've seen in other cases some very large deals done with big pharma or biotech companies. So that's something that we're working towards very hard. At the same time, our personalized off-the-shelf immunotherapy cell lines have been genetically engineered, and we're now working to get them up to speed for GMP production. And the goal is to get those into the clinic later this year, as well as to develop similar immunotherapy cell lines for colorectal cancer, melanoma, and prostate cancer. And those could get into the clinic as early as next year. We're working on the mechanism of action of these things in collaboration with the National Cancer Institute. So those are our major goals right now. We do have the earlier stage program, protein kinase C delta inhibitors, which are very interesting, developed by Doug Fowler and Robert Williams out of Boston University and Colorado State University. We've licensed them in. And the hypothesis there, which appears to be the case, is that cells that are RAS transformed become addicted to PKC delta signaling, that if you block PKC delta signaling, they undergo apoptosis, possibly because PKC delta, when it's active, prevents RAS from being degraded. And so there's a lot of preclinical models that have shown that this is a very promising therapy, but we're working on the small molecules we have to make them a little bit more soluble, better absorbed, and that kind of thing, before we have candidate selection and move forward with our preclinical toxicology studies. So that's probably a year off before.
Yeah, the RAS work is I mean, it's been 3 decades, right? And it's finally starting to crack, and progress is being made. So we look forward to updates on that. And back to you, David, and the MVA platform. And I like to think of GeoVax. I've known the company for many, many years as a company kind of like Bavarian Nordic. And if you want to see what MVA can do, look at what Bavarian Nordic has done throughout its history in terms of smallpox vaccine stockpiling. It's very safe. They have infectious disease and they have oncology. So that's how a good way to frame or comp GeoVax, and I'll use that to now let David talk a little bit about what the company's doing in its lead programs with MVA, including COVID, and that could be stockpiling, and it also includes Ebola, and that could be stockpiling, kind of like what that other group did. So David, maybe you can speak a little bit about your lead programs.
Thank you. Well, our priority program is our coronavirus vaccine program. We don't refer to it as COVID because COVID is a subset of it. But as you know, these variants have continued to emerge. They're in the news. We're seeing the UK, we're seeing the South African, the Brazilian, the Indian. We're currently conducting animal testing including against the South African variant on that. We received funding because of our ability to encode broader groups of proteins into our MVA. We have designed a single dose vaccine, because we get that traditionally in all of our work from MVA, but a single dose vaccine against broader than just the spike protein or the predominant protein. And so it would also, it would cover before the variants emerge. So we have the lead protein is the spike protein, but we have the matrix protein, the envelope protein. We have others that we've encoded in there. We should see results beginning this first half of this year from the animal studies. In the second half, we also have our hemorrhagic fever programs, as you mentioned. We've previously gone through non-human primates, 100% protection, single dose with no adjuvants or boosters necessary for Ebola. We have currently Lhasa, Marburg, and Sudan are all in non-human primate testing being funded by the federal government, so non-dilutive funding. Those results will be coming, some the first half, others into the second half of this year. And we've already had discussions open with us by BARDA about the stockpile program. And as you're familiar with and you allude to, the stockpile program can be very lucrative. The last product that went in there, it was a 10-year deal for $2 billion. And so it's $200 million a year, and the upfront payment was $170 million, and you really have no commercialization costs. So we're expecting that our hemorrhagic fevers will go into that program, depending on obviously successful results. But we also expect will be funded through clinical development by the federal government in the hemorrhagics also. NIH and BARDA we're in discussions with behind our coronavirus program. And that's because dependent on the animal testing, which we hope will be positive, we will then receive sufficient funding to accelerate into clinical development.
And these 30-minute segments go by so quick, but the top, just to kind of close, the topic here was post-COVID, getting back to fundamentals. And starting with the latter first, GeoVax, yes, very active in COVID, but also Ebola, Lassa fever, other infectious diseases, and then they have an entire oncology program as well with MUC1. And if you look historically at a company like GeoVax, incredible technology, had a bumpy road until David got there a couple of years ago and has really straightened or righted the ship of that company. And that's a good way to take a look at it, and good things coming for them this year. Briacel and Bill's group, breast cancer work and vaccines and immune therapies have really built traction over the last 12 to 18 months or so. Take a look back what came out of San Antonio Breast Cancer Symposium in December. Very cool stuff. And looking forward to what he's going to do with his group. Plus that RAS space continues to be very active. And then closing with Actinium and Steve's group. It's a pivotal stage company. That data is going to come, possibly in the next, call it 6 to 9 months. I'm not holding Steve. I'm not asking him to confirm or deny that. We don't know yet. But also they're working in CAR-T. They're in MDS, and they have an antibody warhead enabling technology called AWE that we didn't get a chance to talk about, but something that you should check out on Actinium's website and in their pipeline. But point is, back to fundamentals. There's 3 very attractive oncology related companies, some of us in infectious disease as well. But we'll stop there. Thank you all for participating, and hopefully we'll catch up with you soon.
Thank you.